Dear viewers, this is my next session on the topic bioinorganic chemistry. In my previous session, I have discussed the biological roles of alkali and alkaline earth metals with major emphasis on sodium, potassium, magnesium and calcium ions. In my today's lecture, I will be dealing with the porphyrins, hemoglobin, myoglobin and their roles. So, let us see the lecture plan. First of all, I will be taking the porphyrins and the porphine ring. Then I will be discussing the metalloporphyrins, chlorophyll and iron porphyrins. Chlorophyll is the magnesium containing porphyrin and iron porphyrins are hemoglobin and myoglobin. So, we will be discussing their structure and their roles. We will also discuss the mechanism of oxygen transfer, oxyhemoglobin, oxymyoglobin, further deoxyhemoglobin and deoxymyoglobin, and in the end we will discuss the cooperativity and Bohr effect. Porphyrins are one of the most important bioinorganic compound in which a metal ion is surrounded by the 4 nitrogen of the porphine ring. What is this porphine ring? This structure is showing the porphine ring in which we can see there are 4 pyrrole rings. These are the 4 pyrrole rings 1, 2, 3 and 4, and these are connected by methane bridges. A double bond and a single bond, we can see these are the methane bridges through which the pyrrole rings are being connected. There are 4 nitrogen and in the center we have some space where the metal can be easily fitted. So, porphines are made of 4 pyrrole rings linked together through methane bridges. Porphines have macrocyclic pyrrole system with conjugated double bond. As we have seen in the figure, the system is a large one, therefore, it is macrocyclic. It is cyclic in shape and it is large, therefore, it is macro and cyclic, and they have conjugated double bond. Conjugated double bond means single and double bond occur in alternate positions. These porphines act as tetradentate ligand with 4 nitrogen donor sites. Tetradentate ligand means, tetra word means 4, that means the ligand which donates 4 pair of electrons to the metal ion. Therefore, these are tetradentate and the 4 donor atoms are the 4 nitrogen of the pyrrole ring. 4 pyrrole rings of the porphine carrying substituent other than hydrogen are porphyrins. So, this is the basic difference between porphine and porphyrins. Porphine contains hydrogen and if the hydrogen is replaced by any substituent, then it is called porphyrins. Porphyrins are big molecules, they are cyclic in nature and they are very stable due to the conjugation, that is alternate single and double bond. The size of the central hole which is made by the 4 nitrogen, that hole, its size is estimated to be 0.2 nanometer, which is ideal for accommodating metals of first transition series and lighter alkaline earth metals like magnesium in case of chlorophyll. The metal ions are surrounded by 4 nitrogen of porphine ring in the square planar geometry and the axial sites are available for other ligands. The porphine ring, it makes a square planar geometry because the porphine ring is planar in shape and these 4 nitrogen, they lie on the same plane, therefore, it makes a square planar geometry. Coming on to metalloporphyrins. The complexes in which metal ion is held in the porphyrin ring system is called metalloporphyrin. That means, we just studied the porphyrin rings. If in the center of the porphyrin ring we place metal, the metal can be the transition metal or the lighter alkaline earth metal like I have told magnesium. So, these type of complexes are called metalloporphyrins. These complexes play a vital role in biological systems. The 2 most important metalloporphyrins are hemoglobin, that is the one containing iron, and chlorophyll containing magnesium. Chlorophyll is magnesium complex of porphyrin which plays an important role in photosynthesis. In this metalloporphyrin, in addition to substituents, a double bond in one of the pyrrole ring is reduced to form magnesium dihydrido complex and a cyclopentanone ring is also fixed to the pyrrole ring. So, if we look at the structure of chlorophyll A, This is chlorophyll A. We have two type of chlorophylls, chlorophyll A and B. This is the chlorophyll A. Here we can see this is the porphyrin ring, and in the center of the porphyrin ring, that is the space between the 4 nitrogen, is the metal magnesium. This is the cyclopentanone ring, and here we can see we have one CH3 group. If in place of this CH3 group we place CHO group, then it will result in chlorophyll B. So, the difference in chlorophyll A and B is only this group. If CH3 is present, it is chlorophyll A, and if CHO is present, it is chlorophyll B. Chlorophyll is a green pigment present in the leaves of green plants. It helps in photosynthesis. Photosynthesis is an important redox reaction which occur in nature to convert water and carbon dioxide into the carbohydrates, and oxygen is liberated, and this process takes place in presence of sunlight. So, here if we look at the reaction, we see we have 6 CO2 plus 6 water. In presence of sunlight, with the help of chlorophyll, they converts to the glucose, that is C6H12O6, and the oxygen which helps us to breathe is liberated from this process. Coming on to iron porphyrins, all organisms require oxygen to survive. The oxygen carrier proteins are the proteins which can bind and transfer oxygen from one place to another in the body. Hemoglobin and myoglobin are two most important iron porphyrins because these contain the metal iron in the center of the porphyrin rings. Hemoglobin is an iron porphyrin which is an oxygen carrier, while myoglobin is an iron porphyrin which is an oxygen storage agent. We will see how hemoglobin carries the oxygen and how this myoglobin it stores oxygen. Myoglobin is a small intracellular protein which is present in the vertebrate muscles. It is present inside the cell, therefore, it is called intracellular protein. Its function is to accept oxygen from hemoglobin in muscles and store it until needed for the energetic processes. While hemoglobin is a large intracellular protein which is responsible for the red color of the RBC, hemoglobin is present in RBCs, that is red blood corpuscles. It picks up oxygen in the lungs and delivers it to the rest of the body. Deoxygenated hemoglobin uses some of its amino acid groups to fix up CO2 and transport it back to the lungs. Coming on to the structure of myoglobin. Myoglobin consists of one polypeptide chain, that means globin, that is the protein part, with one heme group. The peptide chain consists of 150 to 160 amino acid residues folded about the single heme group. This is the structure of myoglobin. The heterocyclic ring system of heme is porphyrin derivative. It contains 4 pyrrole groups joined by methane bridges, that is this is the porphyrin ring which has 4 nitrogens. These are the 4 nitrogens of the porphyrin ring. In the center of this, that is in the hole, iron is present in the form Fe2 positive. The iron atom present at the center of the heme is bonded by 4 nitrogen of the porphyrin and 1 nitrogen from the imidazole ring. This is the imidazole residue and it contains 1 nitrogen. The 5th coordination site of iron is occupied by this nitrogen. This is the structure of myoglobin. Here we can see this is the heme group, to which this blue part is the globin part, that is the protein which is attached to this heme group, and this red part is the iron. Coming on to hemoglobin. Hemoglobin is a large protein with a molecular weight of about 60,000. It consists of 4 subunits, each of which contains one heme group associated with protein globin. Therefore, there are 4 heme groups bonded to 4 protein chain. This thing is to be understood that myoglobin contains only one heme group, while hemoglobin contains 4 heme groups. One heme group with its protein chain is called a subunit. So, hemoglobin has 4 subunits. The 4 subunits are similar, but they are not identical. 2 subunits form alpha chains of 141 amino acids and the 2 form beta chains of 146 amino acids. The sequence of amino acids in hemoglobin is different from that in myoglobin. This is the structure of hemoglobin. Here we can see that this blue part and the red part, the folded part, this is the globin. These are the 4 subunits. Alpha and beta units are shown in the red and the blue, while these green parts are the heme parts, that is the porphyrin containing iron in the center. This can be more clearly understood from this figure. Here also we can see there are 4 folded chains. These are called the alpha and beta units, the brown, purple, pink and the dark brown one. This blue part is the heme group, and in the center of heme group this red part is the iron. So, we can see there are 4 heme groups, 1, 2, 3 and 4, each containing one iron atom in the form Fe2 positive. This is the structure of heme group. Again we can say this is the porphyrin, because the side chain are some substituents, they are not hydrogen. So, this is porphyrin, not porphine. So, in the center of the porphyrin is the iron present in Fe plus 2 state, which is connected to 4 nitrogen of the pyrols. Both hemoglobin and myoglobin has 5 coordinated iron second atom. It is bonded to 4 nitrogen atoms from pyrrole ring and the 5th from the protein chain. The 6th position is occupied by weakly bonded water. Then myoglobin and hemoglobin are called deoxymyoglobin and deoxyhemoglobin. This means the iron present in the center of the hemoglobin and myoglobin is 6 coordinated. The 4 coordination sites are the 4 nitrogen from the pyrrole ring, the 5th is from the protein chain, that is the imidazole nitrogen from the protein chain. The 6th position, if the 6th position is occupied by the water, weakly bonded water, then it is called deoxyhemoglobin or deoxymyoglobin, that is oxygen is not present. But if the 6th position is occupied by the molecular oxygen, then these molecules are called oxyhemoglobin and oxymyoglobin. Roles of myoglobin and hemoglobin. Hemoglobin and myoglobin plays very important role in transporting oxygen from lungs to tissues and CO2 in the form of carbonate from tissues to the lungs. Oxygen is inhaled into the lungs at very high pressure when it binds hemoglobin in the blood forming HbO2. This is a complex of hemoglobin with oxygen. The oxygen is then transported to tissues where the partial pressure of oxygen is very low. The oxygen then gets dissociated from hemoglobin and diffuses to the tissues where myoglobin picks it up and stores it until it is needed. Myoglobin has greater affinity for oxygen than hemoglobin. This increases the rate of diffusion of oxygen from the capillaries to the tissues by increasing its solubility. Heme group consists of iron 2 positive, that is ferrous ion, enclosed in a porphyrin ligand. Ferrous has 6 coordination sites. The porphyrin ligand takes up only 4 of the 6 sites, that is by the 4 nitrogen of the 4 pyrrole rings, leaving 2 free binding sites on the opposite side of the metal ion. If free heme is present in aqueous solution, the 2 vacant sites may be occupied by water molecules. The naked heme group binds to oxygen molecule in an irreversible manner when ferrous, that is Fe2 positive, is oxidized to ferric, that is Fe3 positive. What happens, like if the globin part is not present around the heme, then and the 2 sites are free, the 4 are occupied, the 4 are coordinated through 4 nitrogens of the pyrrole ring, and if 2 are free, then the oxygen can bind to them and it will bind in an irreversible manner. Irreversible manner means if it once bind, it will not lose the site, it will be there for always. This oxidation of iron destroys its oxygen binding capacity. Like we can see here, this is the iron 2 present in heme. If it binds with oxygen and water, irreversibly it forms hematine, that is Fe2 positive is oxidized to Fe3 positive hematine. This is the structure of hematine, and hematine is a dimer which no longer acts as oxygen carrier. But what happens in reality, the porphyrin iron is protected due to the presence of hydrophobic protein chain around the Fe2 positive, which blocks the approach of larger molecules to the neighborhood of iron 2 and hence prevent the oxidation of Fe2 to Fe3. The hydrophobic group also prevent the solvation of ions. That means the heme, the ferrous present in heme does not gets oxidized to Fe3 positive, that is hematin, and there is a reversible binding of oxygen with the heme because of the globin part which protects it from the oxidation. Fe2 positive of myoglobin and hemoglobin can be oxidized under certain controlled condition to Fe3 positive, forming met myoglobin of myoglobin and met hemoglobin of hemoglobin. These Fe3 proteins are responsible for the brown color of old meat and dried blood. That means here the heme has been oxidized to hematin. The deoxyhemoglobin. It is a 5 coordinated high spin iron second complex with 4 of the coordination positions occupied by the porphyrin nitrogen. It is always there. The 5th position is occupied by a nitrogen atom from the imidazole side chain of the histidine residue which joins the heme to the protein. That means 4 sites are occupied by the 4 nitrogen from the porphyrin pyrrole ring, while the 5th position is occupied by the nitrogen from the imidazole side chain. That is a protein which is covering the heme. The nitrogen from the imidazole ring, it occupies the 5th position. In the absence of oxygen, the ligand field is weak. So, such 5 coordinated heme complexes of iron 2 plus are always high spin, having the configuration T2G4 and Eg2. Therefore, these 5 coordinated complexes are paramagnetic. In this high spin state, Fe2 positive has larger radius than in low spin Fe2 state, having the configuration T2G6. In this, all the electrons are paired because of repulsive effect of one Eg electron occupying the dx square minus y square orbital, which is directed towards the 4 nitrogen atom of the porphyrin ring. This can be understood very clearly from here. In this figure, we have 2 complexes, the high spin complex and the low spin complex. In the high spin complex, this is the ferrous ion containing 6 electrons in the valence d orbital. Here we can see that 4 of the electrons they enters the T2G orbital, while 2 enters the Eg orbital. Here the electrons are unpaired and this is paramagnetic in nature. While in case of low spin complex, we can see that all the 6 electrons they are paired and they are present in the T2G orbital, while Eg is vacant. So, in case of ferrous high spin complex, the electrons are present in Eg orbital. Now, this causes the repulsion with the dx square minus y square orbital, and hence the size of this ferrous ion is larger, while in case of low spin complex, the Eg orbital are vacant and there is no such repulsion because dx square minus y square orbital is empty and the size of this iron is smaller than the size of the high spin complex. This is the structure of deoxyhemoglobin, and here we can see that the iron it lies above the plane of the 4 pyrrole. We can see that the iron lies above the plane of the 4 nitrogen from the pyrrole ring. The estimated iron nitrogen distance is larger than the size of the central hole of the porphyrin ring. Therefore, this iron it lies above the plane of the 4 nitrogen atom by about 70 picometers to give a square pyramidal arrangement. So, we can see that here the arrangement is square pyramidal, a square formed by these 4 nitrogen, and since this iron it lies above the hole, so it makes geometry of pyramidal. It makes a geometry of pyramid, therefore, this is a square pyramidal geometry. Coming on to the oxyhemoglobin. When oxygen binds to the 6th position, iron becomes coplanar. In such a situation, oxygen gets coordinated to iron second. That means the iron which was above the plane of the 4 nitrogen, now it gets into the hole of the 4 nitrogen and the structure becomes coplanar and it becomes easy for oxygen to get coordinated to the iron. The ligand field becomes strong, resulting in the spin pairing, giving a low spin, that is T2G6 structure, which we have just now discussed. The resulting complex is a diamagnetic and iron nitrogen bond. Distance is approximately same as the porphyrin hole. Therefore, the iron it gets fitted into the hole. Therefore, iron becomes coplanar. The release of strain energy of the square pyramidal complex compensates the loss of the spin pairing energy in going from 5 coordinated deoxyhemoglobin or deoxymyoglobin to the 6 coordinated oxyhemoglobin or oxymyoglobin. Therefore, the coordination of oxygen results in dropping of the iron in the plane of the heme group. So, this can be understood from this figure. Here we can see that the iron, it lies in the plane of the 4 nitrogen of the porphyrin ring. This lies in the plane of the nitrogens because the size has decreased. When it has coordinated to oxygen, now it is low spin complex and its size has decreased, so it falls into the hole of the porphyrin. Coming on to cooperativity. If we see, the net result of this interaction in hemoglobin is to increase the affinity of oxygen of the second heme site and so on. As one iron binds an oxygen molecule, the molecular shape changes to make the binding of the additional oxygen molecule easier. This phenomena is called cooperativity. Cooperativity enables hemoglobin to bind and release oxygen more effectively. This can be understood from this figure. Here we can see that this is Fe2 positive with a large size because it is present in high spin. This is the porphyrin hole, and here also we have the porphyrin hole, but here the size of this iron, now this is 6 coordinated, the 6th coordination site is occupied by the oxygen, the size has decreased and it is low spin complex, and since the size has decreased, this iron falls into this porphyrin hole, whereas here the iron, it is above the plane of the porphyrin hole by about 70 picometers. So, this reaction keeps on taking place, like when it combines with the oxygen, it falls into the hole, and when the oxygen is released, the size again increases and it stays above the hole. Functions of myoglobin, hemoglobin and cooperativity. The main function of hemoglobin is to bind oxygen at high partial pressure of oxygen in the lungs and then carry it through the blood to the tissues where myoglobin picks up oxygen from hemoglobin. In the lungs, where the pressure of oxygen is high and much oxygen is bound, the affinity of heme for oxygen becomes very high. It effectively loads up with as much oxygen as possible. When the hemoglobin reaches the cell where the pressure of oxygen is low, oxygen begins to dissociate from the complex, and now the myoglobin picks up all the oxygen.